Hello there, future doctors. Welcome back to yet another video by Best Medicine for GCSE AQA Biology. Today we'll be continuing. In this video, we'll be continuing with the chapter that we started, which was digestion, but we have also included enzymes into it. So we have so far finished different types of biological molecules and their food tests. Very, very important information. And today we'll be starting with enzymes. So what are enzymes? Remember, when we were doing proteins, we said one example of proteins is to make enzymes, right? So enzymes are three-dimensional proteins that help catalyze chemical reactions. What is catalyzing? Catalyzing essentially means to be able to allow a chemical reaction to occur in an easier way, in a way that requires lesser amount of energy, right, and can occur at for example, in the body's normal conditions. So that's what is catalyzing. When you study chemistry, you learn that catalysts are able to decrease something called activation energy in chemical reactions. And so enzymes are kind of like biological catalysts because they allow reactions to occur at normal body conditions of plants, of animals, of humans, etc. So, as catalysts, they help speed up reactions in the body without themselves being used up. Now, there's two different kinds of reactions in the body. Number one, we have the anabolic reactions. What are anabolic reactions? Anabolic reactions are those reactions that help in making up stuff from smaller stuff. So, these are building up reactions. For example, you could have something like a triangle plus a rectangle, and then you get this resultant shape, right? Something like this. And so you've gone from 2 to 1. That's an anabolic reaction. If you're trying to build up muscle, that's anabolism, right? If you're breaking down muscle, then that's catabolism. So for example, if you were to break down something, let's say here's a thing that is made up of some red part and some green part, right? And this undergoes a chemical reaction and you get this, right, and you get this, then this is catabolic reaction. Okay, an example of catabolism or catabolic reactions are digestion, right? Digestion is a catabolic reaction in which you break larger molecules into smaller molecules. So the sum of all of these reactions, all anabolic and catabolic reactions, is referred to as metabolism, metabolic reactions, okay? Metabolic reactions include both anabolic and catabolic reactions, and the sum of all catabolic reactions and anabolic reactions in the body is called metabolism. So metabolism is a very popular word that they throw at you in the books, and they don't actually tell you what this means. So this is what it means, okay? So now we go into enzymes. How would enzymes conduct these reactions of anabolism and catabolism? How will they be able to do this? Well, the answer to this is that enzymes are designed in a very specific way to allow the catalyzing of these reactions. So remember, enzymes are proteins. Enzymes are three-dimensional proteins, so they have a very specific shape which allows them to bind the substrates and allow the catalysis. So how does this work? Well, remember when we were doing proteins, we said that proteins are biological molecules made up of many amino acids connected together, right, forming a polypeptide chain. So that's a polypeptide chain, and let me just draw this as a line, and what would happen is that to form a three-dimensional structure, this would fold onto itself. So here's it's folding, and it's got this specific shape, right? It kind of looks like this. Let me just show you, right? That's a protein. So this, in our case, is an enzyme. In our example, this protein is an enzyme. So how will this enzyme work? Well, what it has is that it's got a special place that is able to bind the substrates that it's supposed to catalyze the reactions for. And that place is complementary in shape for that substrate. It is specific in shape for that substrate. And that is, in our example, this shape here, right? You see this? It seems like anything that is rectangular, can very easily fit into this. So this place is called an active site, and that's a very important term, okay? Very, very important term. I encourage you to use as much of this word in your answers related to enzymes as you can. So active site is the site of the three-dimensional protein enzyme that is able to catalyze reactions in which the substrate is going to bind. So what is a substrate? A substrate is the reactant, right? A substrate is, for example, this one here is the substrate. This one here is a substrate in these two examples, okay? Before the reaction occurs. So in this case, in our example, you could have a substrate that kind of looks like this, right? Complementary in shape to the active site. And so this is our substrate. And when it fits into the enzyme, it forms something called an enzyme substrate complex, okay? Enzyme substrate complex. And once this is formed, the reaction can occur. So in this case, our example, maybe we can make this into a catabolic reaction. Remember, a catabolic reaction is a reaction in which you break down stuff. So what would occur as a result is that you would get the substrate being broken down. Maybe one of the product kind of looks like this, right? And the other product kind of looks like this. So we've got two products here, plus we've got the enzyme. The enzyme does not change at the end of the reaction, just like catalysts don't in chemistry. So the enzyme is still there while we've got these two products. So that's a catabolic reaction. You could also have an anabolic reaction if you want me to make that one. It kind of would look kind of like this, that you've got an enzyme, right? And then you can have two substrates here, maybe a yellow one and a green one, right? And then what would happen as a result is that you get this product that is a combined total of the two substrates, right? And the enzyme again would be unchanged. Understood? So how do we define this particular mechanism? How do we define how enzymes work? Well, we do it with using a model, and that is called the lock and key model or lock and key hypothesis. The lock and key hypothesis was proposed by some scientists, and it said that that enzymes and substrates, they kind of work with each other like a lock and a key would work with each other. Remember, a key is very specific for a lock, and it is able to unlock the lock and itself remain unchanged. Similarly, an enzyme, right, is specific to the substrate. So the lock and key model of enzymes states that the enzymes have specific active sites that fit perfectly is a key word here, with substrates, like a lock and a key. Only the substrate with the correct shape can bind to the enzymes, and if they're not the correct shape, then they cannot bind to the active site. And therefore, and as a result, it would undergo a chemical reaction. The enzyme remains unchanged at the end of the reaction, just like the key. So the key in this case is the enzyme, while the lock is the substrate, because key is unchanged, right? Remember? There are better, more accepted models when you learn A-level biology, but really this is what you need to know for GCSEs. Now, something very important that I want to mention here is that remember that this is a 3D protein. So if this is a three-dimensional protein made up of polypeptide that is joined by weak bonds to, I'm not talking about peptide bonds, but some weak intermolecular forces here to form the 3D structure, then you can easily break these bonds. If you heat it, we also discussed this in the previous video, if you heat it, then that's going to increase the kinetic energy. Or if you increase or decrease the pH, acidity, extreme acidity or alkalinity, then it can result in the changing of the three-dimensional structure. Now, if three-dimensional structure changes, then the active site is lost. Let me repeat this. The active site is lost. That's a key word that you want to put in your answer when they ask you, why does the enzyme stop working at high temperatures? Or they can give you an example of a graph in which they show you some data, and you notice that at high temperatures there's no reaction happening, and they ask you, why is there no reaction happening at such a high temperature? And you would say, because at high temperatures the kinetic energy of the molecules increases, which causes the protein, the enzyme, to be denatured. When the enzyme is denatured, then you lose the active site. When you lose the active site, the substrate can no longer fit into the active site, enzyme substrate complexes cannot be made, and therefore no reaction occurs when the enzyme is denatured. Okay, so effect of temperature on enzyme function. We'll have to do some graphs of this so that you can get familiar with any data that you see on the exam. So let's just read this through again as a revision guide. Generally, increasing temperature would increase kinetic energy, right? Well, in chemistry, when we increase temperature, normally that increases rate of reaction. So what's going on here? Well, over here as well, it normally increases rate of reaction, because when you increase temperature, it would increase the frequency of collisions between the substrate and the enzyme, and it also increases the number of successful collisions, right? Due to more particles having the activation energy. This is a little bit of chemistry involved here. So, generally, increasing temperature would increase the reaction. However, if you keep on increasing temperature, right? If you keep on increasing temperature too much will unfold the protein, and the active site will be lost because there's too much kinetic energy. So then the enzyme is said to be denatured, and the active site is lost. Understood? Now, something that I want to mention here is that every enzyme would have its own optimum temperature. Okay? Different enzymes have their own optimum temperature. What is an optimum temperature? An optimum temperature is the temperature at which an enzyme would work best. For example, most of human body's enzymes would work best at the human body temperature, which is how much? 37 degrees Celsius, right? So most of the human body's enzymes would work best at 37 degrees Celsius. If you increase the temperature too much or decrease it, that would cause the enzymes to not work very well. That is why fever is bad for us. Okay, well, we've looked at how increasing temperature would increase the kinetic energy and result in denaturation of protein, and resultantly the active site is lost. But decreasing temperature does not follow the same logic, because when you decrease temperature, you're not really causing the molecules to shake violently and denature the protein. So what would happen if you decrease the temperature? Well, decreasing temperature does not denature an enzyme. However, it just decreases its function, because there is no kinetic energy, right? Very less kinetic energy means there's less frequency of collisions and less number of successful collisions, because less particles have activation energy. So the function decreases when you decrease the temperature away from optimum. But if you increase the temperature away from optimum, then the enzyme can become denatured and the activity can drop to completely zero. So let's make a graph here and try to make sense of this. Okay, so on the y-axis, you want to have the enzyme activity, right? That's the thing that you want to change. That's the thing that you want to observe. And on the x-axis, you're going to want to have the temperature. So how does increasing the temperature affect the activity? Remember, always, the independent variable, you always write it on the x-axis, and the dependent variable, you always write on the y-axis. So let's say 37 degrees Celsius is the optimum, and 37, let's say, is around here. 37 degrees Celsius, right? So here's what occurs. At 37, the activity is maximum. But as you increase the temperature too much, eventually you see the activity decreasing to zero. So that's the thing there, and this is the kind of graph that you should expect for an enzyme function versus the temperature. So notice that when you decrease the temperature, the activity does not drop to zero. When you increase the temperature, the activity does drop to zero, and this is irreversible. And the reason for this is because, again, when you increase temperature, there's a lot of kinetic energy, which means that the particles, the enzymes, are denatured. They lose the active site, so enzyme substrate complexes can no longer be formed, and the reaction cannot occur. But if you decrease the activation energy, then it does not denature the enzyme, it just decreases its function, because there's less kinetic energy, which means there's less frequency of collisions and less particles have activation energy. Okay? Well, that's for temperature. Well, what about for pH? What about for pH? The pH, it's going to be a little bit similar, but a little bit different. And I think the pH graph is a bit easier. Basically, effect of pH on enzyme function is similarly to temperature. Every enzyme has its own optimum pH, right? So it would have its best pH. For example, some enzymes that work in the stomach would work best at acidic environments. Some that work in the small intestine would work slightly at slightly alkaline environments. Some that work in red blood cells would work best at neutral pH, right? So different optimum pHs for different enzymes. Now, if we go too far away in pH on either side, if you increase the pH away from optimum or decrease the pH from the optimum, it would denature the enzyme. Okay, there's no two words to this. It would denature the enzyme, regardless of if you increase it too much or decrease it too much. All right, so let's have a look. So on the x-axis, we would write our pH. That's the independent variable. And then on the y-axis, we would write the activity of the enzyme, right? Maybe the rate of reaction. And let's say our normal pH is 7.4, which is the normal pH of blood, and optimum pH. And over here, the activity, if you see, goes to 0 when we go to either side. It goes to 0 because the enzymes are denatured. The active site is lost. The enzyme substrate complexes can no longer be formed because the substrate does not fit into the active site and the reaction cannot occur. Okay? So that is the effect of pH on enzyme function. Very quickly repeating this, for temperature, if you increase temperature, it will denature. If you decrease temperature, it will not denature, and this is reversible because you don't lose the active site. If you increase the temperature again back to optimum, it's going to go up. For pH, it will get denatured and it will no longer return back, right? This is irreversible. And that finishes our section on enzymes. So I have a question for you guys. What is your favorite enzyme and why? I am very curious about this one, so please let me know, okay? And quote your nicknames, please. I would really like an answer to this. So, the legendary digestive system. We start off with some memes. My face when someone walks in that I am supposed to be on a diet with. Yep. Who has failed diets before? Not me. Touch my cake and I'll cut you. This boy looks serious. Okay, so digestion. Digestion is, of course, one of the reasons why we are alive and one of the reasons why we love food so much, because we're able to digest it, right? Break it down and get the nutrients from it. So digestion is a process of breaking down food into smaller pieces, both mechanically, so that it can be swallowed, as well as chemically. Chemically would mean, just like we did earlier, maltase and amylase, right? Breaking the bonds, the chemical bonds between them. And eventually, once you have the little molecules, like amino acids and glucose, you can absorb them. So in this chapter, we will learn mechanical digestion, which is a very brief little topic, chemical digestion, movement of food, and absorption of food, and together with all the other organs, that are associated with them. So number one, we've got the mouth. The mouth, also referred to as the oral cavity, contains teeth, right? Starting from the front to the last 8, we have the 8 incisors, so 4 on the top, right, and then 4 at the bottom, and these are 8 incisors, and 4 canines. Canines are evolutionary inheritance from carnivore genes. 8 premolars, so canines help us really cut meat, and then 8 premolars and 12 molars, and then these basically all what they do is they help us chew the food. Now in AQA you would not be directly questioned about any of the teeth's names, okay, and you would not be questioned about the function generally of each of the teeth. However, you should know that in mouth you have mechanical digestion, right? Physical also called physical digestion. The mouth also contains salivary glands, right? The mouth also contains salivary glands. We actually have 3 of them if you ever 3 major salivary glands. If you have a person, right, we have some glands here, okay, and we have some glands here and underneath the tongue, and we have a gland here, and all of them secrete saliva into the mouth. So salivary glands, they secrete saliva, which contains, number 1, mucus. Mucus is a watery substance. Mucus, basically what it does is that it helps mix chewed food into small boli, also called a singular bolus, to be swallowed. So it basically helps the food to be mixed together and help make a small bolus. Number 2, the enzymes contain salivary amylase. That's very important, okay? Salivary amylase. Do you remember the amylase enzyme from the previous video? Amylase is the enzyme that helps break down starch, right? So amylase is the enzyme that helps break down starch into maltose, which will then be digested later into glucose in the intestines. And number 3, the organ that is present in the mouth is the tongue. Tongue will help mix the chewed food together with saliva. It also contains taste buds, which essentially are chemical receptors that detect the taste sensation and generate nerve impulses. And again, these impulses are carried to the brain to be interpreted by a sensory neuron, which is part of a nerve. More on this in its own chapter. So yeah, that essentially is the mouth. The mouth does 2 types of digestion. Number 1, mechanical or physical digestion by the teeth and the tongue. And number 2, chemical digestion by salivary amylase, right? It also contains mucus-producing salivary glands that are able to mix the food into small boli to be swallowed. It contains a tongue that helps chew the food and be able to test the taste from the chemicals in it. Then once it is swallowed, it goes into the esophagus. So where's the esophagus? Essentially, when the food is swallowed, it enters this space behind the mouth, and this is called the pharynx. And the pharynx is connected in front to the trachea, which is the airway, and at the back with the esophagus. On top of the trachea, you have a layer of tissue that blocks the trachea when the food is swallowed. So whenever you're swallowing some food, it's moving down like this. The trachea will be blocked by something called epiglottis, okay? It's called epiglottis. Epiglottis will block the trachea so that the food that you're chewing will enter into your esophagus and go down, okay? Sometimes when we're not paying attention or trying to talk while chewing or swallowing, a little bit of food can go into trachea, and that can result in a very strong cough reflex. So if that happens, and I'm sure you guys are familiar with it, then don't be shy to cough, okay? The first thing and the most important thing to do in that situation is to cough as hard as possible to remove the food out of this. Okay, so that's the esophagus, and this is where the food goes in. So what is the esophagus? What does it contain? The esophagus is the organ that contains circular and longitudinal muscles which help perform wave-like contractions to push food along into the stomach. This wave-like movement is called peristalsis. Very important, okay? Peristalsis. Peristalsis occurs throughout the gastrointestinal tract, the esophagus, the stomach, the small intestine, the large intestine. So how does peristalsis work? Let me just quickly show you, okay? So here's the esophagus, right? And then here is the food that we have. Maybe I can just put it here. Now, what the esophagus will do is that it's got these 2 sets of muscles here. One of them are circular muscles, and the other one are longitudinal muscles, okay? Now, when it wants to push the food along, it will contract the circular muscles from the top of the food. Okay, circular muscles will contract from the top of the food, which causes the esophagus to move inwards, kind of like this, okay? So it will go like this and be able to push the food from the top. Okay? While below the food, right? Below the food, it causes the contraction of longitudinal muscles, which causes the esophagus to dilate. All right? Allowing the food to go down much more easily. And when it reaches over here, again, in this place, then again, it would cause the circular muscles to contract, pulling the esophagus inwards. So maybe we can draw a new layer here, okay? And longitudinal to relax in front of it, and the food will go down. So this is a wave-like contraction that would keep on occurring from the top of the food. You contract the circular muscles, and below the food you contract the longitudinal muscles to allow it to be pushed down into the stomach. Okay? And this movement is called peristalsis. Not only does it occur in the esophagus, it also occurs in the stomach, and it occurs in the small intestine and the large intestine to push all the food and digested contents along the gastrointestinal tract. So as food is pushed along, eventually it will reach the stomach, and so on. So again, this is another diagram of peristalsis. Remember, longitudinal muscles, they help dilate, while the circular muscles, they help contract from the top. Okay? And that's called peristalsis, pushing the food downwards. So question, define peristalsis, 2 marks. Let's see who can get full marks on this one, and code your answers with nickname and end the question, please. Okay? And next up we have stomach. The stomach is the organ that is next after the esophagus in the gastrointestinal tract. Now, how does the stomach help in digestion? Well, there's a couple of things. Number one, the stomach contains these glands, like glandular tissue in its wall. So here's the wall of the stomach. I've just taken the wall of the stomach here. And remember, the food is here, right? The food is in the lumen of the stomach. So here's the food, and we want to break this food down. So what does the stomach do? Well, the stomach has these pits in which we've got glands. These glands, they help secrete some enzymes. A very important enzyme is pepsin. Pepsin is an enzyme that comes in here. The other thing that the stomach does is that it helps secrete hydrochloric acid. So these glands are secreting 2 things, pepsin and hydrochloric acid. Hydrochloric acid will digest the food into a liquidy paste called chyme. Okay? So let's write this down. Digests the solid food into an acidic liquid called chyme, which will then enter the small intestine. The other thing that occurs here is that these glands, they secrete something called pepsinogen. Pepsinogen is an inactive form of pepsin. Remember, pepsin is a protease. So pepsinogen is secreted, and pepsinogen converts into pepsin, which is active, due to the acidic pH. Because pepsin works better at acidic pHs, it does not work at neutral pH. So pepsinogen is inactive. And then it would function as a protease, and it would break down the peptide bonds in any proteins that we eat. So 2 features so far. Number 1, stomach, it contains walls which have glands, and these glands are able to secrete hydrochloric acid, most importantly, that is able to digest the solid food into an acidic liquid called chyme. You know what? Hydrochloric acid also helps in, it breaks down any swallowed bacteria. Breaks down any swallowed bacteria as well. So it kind of provides this indirect immunity to us. The third thing is that the stomach contains these walls of muscles. Just like in the esophagus, we had a circular layer of muscles and longitudinal layer of muscles. In the stomach, we also have circular layer and longitudinal layer. When the circular contract, they help push the food inwards. Longitudinal contract, they pull the wall outwards. And this helps generate these wave-like contractions called peristalsis, which will help push the acidic chyme into the small intestine. Okay? So I know it's a little bit much, but you can repeat these videos. That's why we have videos, right? You can repeat these videos as much as you need to get familiar with this. So stomach, it is the organ adapted for chemical digestion. It secretes HCl to help digest the food. Also, just a little bit of an extra information is that the stomach, it protects itself from the HCl as well. And the way it does that is that the stomach epithelium secretes an alkaline mucus, which protects itself from being digested from the HCl. Secretes pepsinogen. Pepsinogen is the inactive form of pepsin, and it is converted to pepsin when it enters the acidic lumen of the stomach. And then remember, pepsin is a protease, so it helps break down proteins into amino acids. And number 3, the stomach contains these circular and longitudinal muscles that help in peristalsis, in pushing the food along. It contains circular muscles, longitudinal muscles to help in peristalsis and churn the food with the acid as well as push it along. Now, food entering the stomach is digested and becomes a viscous liquid called chyme. The chyme then enters the small intestine for further digestion. Okay? So, the small intestine. The small intestine has got 2 functions. Number 1, it is able to digest the food. Number 2, it is able to absorb the food. The absorption part we already did previously in the cells and exchange chapter, but we'll repeat a little bit on it in this chapter as well. But most importantly, we're going to talk about digestion. So one of the things is that the small intestine is able to generate its own enzymes, its own secretions. So it's got its own enzymes, the intestinal enzymes, that help break down the food. Number 2, it receives enzymes from the pancreas. Not only does it have its own enzymes, it receives enzymes from the pancreas as well, so pancreatic enzymes. It also receives bile, which comes from the gallbladder. So how does all of this work, and how does it look like, and how does it all tie up in a human being? Well, remember, a human being, right, has a head and everything, right? And then you've got the arms and everything, and then you've got the liver and stuff. So liver is situated, and I'm going to start with the liver. I know it's a little bit out of proportion, but the liver is situated on the right upper quadrant of the abdomen. Right upper abdomen. Now what the liver does is that it secretes something called bile, and bile helps in digestion of fat. So liver secretes something called bile, and that bile is going to come in and be stored in the gallbladder that is situated just below the liver. Okay, that's the gallbladder. Now at some point this is going to enter the small intestine where the food is coming to. Let's have a look at the stomach. The stomach is present on the left side, generally, of the human body, right? Kind of like this. And then the stomach is making the chyme, and that's going into the small intestine. So this is the stomach that we've already finished, and then here's the small intestine. Now, at the same time, we also have an organ situated underneath the stomach, connected to the small intestine, and that is called a pancreas. So we also have an organ over here, and that kind of looks like this, right? And that's called the pancreas. The pancreas is also connected to the small intestine. Now, the gallbladder, its duct, which is bringing bile, is called the bile duct, and it merges with the pancreatic duct to eventually deposit its material as well to the small intestines. Alright, so let's start with the small intestine and its functions. So remember that we had food that came in through the esophagus into the stomach, and the stomach secreted HCl and pepsin to digest the solid food into chyme. Now this chyme, which is a liquid, right, enters into the small intestine. Because chyme is acidic, pancreas secretes some alkaline juices to be able to neutralize this. Now, what is the role of small intestine in digestion? Well, small intestine secretes many different kinds of enzymes. For example, small intestine will secrete carbohydrates. Carbohydrates will break down carbohydrates in the food. It would secrete proteases, right? It would have proteases, which will break down proteins into amino acids. It will also have nucleases, that would break down nucleic acids, for example, DNA, RNA, although this is usually not tested. And it would have lipases. And remember, lipases break down lipids, and lipids are made up of fatty acids and glycerol. So every time a lipid is digested, lipase will break down a fat molecule, a lipid, or a fat molecule into 3 fatty acids, right? 3 fatty acids and 1 glycerol molecule. And fatty acids are used in aerobic respiration to release energy during exercise. Proteases, you're already well aware of, will break down proteins into amino acids, which will then be absorbed and help make proteins in the body. Carbohydrates will break down sugars into simple sugars like glucose, which will help in aerobic respiration. Now, let's talk about the pancreas. What is the pancreas role in all of this? Well, the pancreas, apart from its endocrine function of being able to secrete insulin and glucagon, the function that we want to talk about today is that the pancreas is able to secrete pancreatic enzymes. Just like the small intestine has enzymes like carbohydrates, proteases, nucleases, lipases, pancreas also has pancreatic carbohydrates, right, which will break down carbohydrates. Amylases, right, it would have amylase, which is a carbohydrate, and it would have proteases, right, and lipases, etc. And all of those enzymes are coming into the small intestine through this pancreatic duct. On top of this, the pancreas secretes alkaline juices, which will help neutralize the acidic chyme that is coming in. So alkaline juices will help neutralize the chyme so that the enzymes that we have here can also work. Remember that the chyme is very acidic, and you don't want the enzymes to be exposed to a very acidic environment. These enzymes are not suitable to the acidic environment. They will denature, and so we want to neutralize the chyme before these enzymes in the small intestine and the pancreatic enzymes can work. So the pancreatic lipases and carbohydrates and proteases, as well as the small intestinal enzymes, would only work when the pH is close to neutral or even slightly alkaline, which is why the pancreas secretes alkaline juices to help neutralize the acidic chyme to be able to have an optimum pH for all these enzymes. The pancreas also has an additional role, which is not in this chapter. That role is that it's got these groups of cells in it that are able to secrete something called insulin, right, and glucagon. These two things are not connected to the digestion chapter, so we'll leave this as is. What about gallbladder and liver? The liver, which is here, liver makes bile. Okay, one of the functions of livers is that it makes bile. What does bile do? Bile helps emulsify fats. Okay, it helps emulsify fats so that they can be better digested by lipases. Okay, so bile is made in the liver, but it is stored in the gallbladder until we eat some food containing fats. When we eat foods containing fats, then the gallbladder will release the bile into the small intestine through the pancreatic duct, and that will help break down the fats in the. So again, bile helps in emulsification of fats. Emulsification of fats, so they can be broken down by lipases. Bile is also very dark in color, dark orangish in color, so it helps give feces its color. Writing in bracket also gives feces its color. Some people, they can get gallstones. Have you heard of gallstones before? Gallstones are stones that can form over here in the bile duct or over here in the gallbladder. And if these stones are formed, then do you think that bile will be able to reach the small intestines? No, right? So if bile cannot reach the small intestines, then do you think that we'll be able to digest or break down fats easily? No. And do you think that we'll be able to have color to the feces? No. So people who have gallstones and stones in the bile duct, their feces are very gray in color, and they have very severe pain on the right upper quadrant. That's where the gallbladder is. And they're not able to digest fats very easily. Okay, so these are the functions of all of these organs, and on an image, you're expected to identify each of them. Okay, you're expected to identify each of them. So that's very important for you to be able to identify that that's the pancreas, that's the stomach, that's the liver, that's the gallbladder, and just underneath, we will have the large intestine. Okay. For the absorption part, you can refer to the previous video in the cells and exchange chapter. Okay. Alright, so let's read the notes very quickly. Digestion, using enzymes such as carbohydrates for sugars and proteases for proteins and peptidases and lipases for lipids together with bile and nucleases for nucleic acids. It helps the food. The small intestine helps digest the food. So carbohydrates will break down complex sugar into glucose, which is absorbed into the bloodstream. Proteases, peptidases will break down proteins into amino acids, which are then absorbed. Lipases break down lipids into fatty acids and glycerol. Okay, and fatty acids and glycerol actually don't enter the blood vessels, they enter the lymphatics. We've done this before. Lymph vessels take fatty acids and glycerol, while the blood vessels, they take the sugars and the amino acids. Absorption is facilitated by 3 adaptations. Number 1, we've got adaptations that increase the surface area to volume ratio for example, having folds, villi, and microvilli. It's also well supplied by blood vessels and lymphatics. Lymphatics will take away the fatty acids and glycerol, and blood vessels will take away the amino acids and glucose. And they have numerous mitochondria because we want to have energy for active transport of both glucose and amino acids. Okay? All of this has been previously explained. Pancreas we learned today. So pancreas makes pancreatic enzymes that enter the first part of small intestine and help in digestion. These include pancreatic amylases, lipases, carbohydrates, proteases, etc. And remember that these work well in the alkaline environment. So pancreas also secretes alkaline juices to be able to neutralize the acidic chyme that is coming in from the stomach. If you don't neutralize the chyme, then these enzymes would not have the optimum pH to work. Pancreas also makes hormones like insulin and glucagon, but that is covered in the homeostasis hormonal coordination chapter. Liver and gallbladder. Liver makes bile salts. Bile is made up of 2 things. Bile salts, although not specifically stated in AQA, but bile is made up of 2 things. Bile salts, which help emulsify fats, and bile pigments, which give feces their color. And gallbladder is a sac-like organ, which really has no important role except for storage of bile. Okay, so you store the bile here until you eat the fats. Next up, you have large intestine. So where is the large intestine and how does it work? Let me just redraw everything, okay? Because identification is very important. So again, this is the esophagus, the stomach, okay? The stomach. And over here, you got the liver, which is kind of like the triangle. And then underneath the liver, you got the gallbladder, right? And here's the bile duct, which is coming into this particular pancreas here. And so on. So the small intestine has a lot of branches here and so on, and then eventually it will conclude over here with a large intestine, which is very thick. And this large intestine eventually concludes as the rectum and the anus. The last part is called the rectum and then the anus. So large intestine has its own functions. Remember, small intestine is able to do digestion and absorption of food. The large intestine does not do either of those functions. It is not involved in digestion of food, and it is not involved in absorption of food. Rather, the large intestine is absorbed in absorption of water and mineral ions. So any excess water here to make feces, this will be absorbed by the large intestine. Any salts here, like sodium, right, chloride salts, etc. These will also be absorbed by the large intestine. All right, so they help essentially prepare or finalize the feces. So if somebody asks you where does the feces get made, you would say it's the large intestine. This is where the absorption of water and salts occurs. So, large intestine absorption of water and mineral salts takes place here. No absorption of glucose, amino acids, fats. Eventually, the unabsorbed fecal material matter is transferred to the rectum and exits through the anus as feces. Okay? And to conclude this particular session, we have a required practical. We love this part, right? Let's do some science. So we have an experiment. This time we test range of different pH solutions on digestion of starch by amylase. Remember enzymes, right? Enzymes would work differently at different pHs. So how does the starch get digested by amylase at different pHs? And that's what we're going to test. Now, generally in AQA Biology, the experiments are easy and very easy to understand. But this one is a little bit tricky. So you might want to repeat this portion of the video a couple of times. But yeah, really what we're going to be testing about is the different pH solutions and their digestion of starch by amylase. So remember, different pHs would mean that amylase, which is an enzyme, is going to be influenced. If at optimum pH, amylase will work best, while at away from optimum, it may even denature. So what are the required apparatuses for this? These are a beaker, a test tube, tripod, and gauze and Bunsen burner, amylase solution, starch solution, buffer solution, which is used for adjusting the pH. pH meter to check the pH, stopwatch, thermometer, spotting tile, iodine solution, syringe, and a dropper. So how are we going to use these? Let's have a look. Basically what we do is we try to measure the time that it takes for the enzyme to become denatured, right? How quickly does it become denatured at different pHs, okay? So we get a range of different test tubes at different pHs, and we mix the starch and the amylase solution in them. So for example, we get a 4 pH, right, and a 5 pH, and 6, and 7, and 8, and maybe even a 9 here, okay? And we place each of them in a water bath that we have set up using the Bunsen burner, the tripod, the gauze, the beaker, and the water, and the thermometer. And maybe we standardize the temperature at 40 degrees Celsius. So every time we place the test tube inside the water bath, we start and mix the contents together. We start the timer. And we measure the time every 30 seconds. And every 30 seconds, what we do is we take, using our dropper, we take a drop and you put it onto the tile. Now normally, because we added starch solution, it should turn blue-black. Right? Before it's digested, it should keep on turning blue-black. So for example, we can record this on a table, something like this. We can say, okay, the pH that we used, and time to stop turning blue-black. Now remember, amylase will break down starch. If starch is gone, do you think that the dropper's drop will turn the tile blue-black or not? Of course not, right? The iodine solution that is present on the spotting tile will not turn blue-black if the starch is digested. So let's start with a pH that I consider as optimum, maybe 7, right? So at 7, you would have the least amount of time that it takes for it to stop turning blue-black, maybe 60 seconds, right? 60 seconds. So you took a sample at 30 seconds and it turned blue-black, but at 60 seconds it didn't turn blue-black. Then maybe we start decreasing the temperature away from this optimum, and we get 6, right? And maybe it's 90 seconds, and then 5 is 150 seconds, and then 4 is like 300 seconds, and so on, right? And 8 is similar, 90 seconds, and 9 is 150 seconds, and so on. So we get these numbers, and we find out that, okay, the starch is being digested best at 7. So that must be the optimum. The optimum pH, while the other ones are the ones that are away from optimum, okay? And that's the experiment, and they can give you this experiment, and they can ask you multiple questions on this, different kinds of questions. Now, in this experiment, the things that we have controlled to prevent them from influencing the experiment are the temperature, right? We standardize the temperature at 40 degrees Celsius, so it does not influence the activity of the enzyme. We also standardized the concentration of starch and the amount of starch that is used. And we also standardized the concentration and amount of amylase that was used. So those would be our control group. Variables that we have controlled, the constants. So controls would be the temperature, right, the concentration amount of starch solution, and the concentration amount of amylase solution. The independent variable is the pH. That's the thing that we're changing. Remember, when we draw a graph, we write the independent variable on the x-axis, and the dependent variable, we write it on the y-axis, right? So the dependent variable in this case was time for iodine solution to not change color, time for starch to be digested. And yeah, hooray! So that's the difficult, required, practical. Hopefully it won't show up. And yeah, science. Yep, who's feeling the rush for understanding some science? I am. And yeah, so if you have any questions, let me know in the comments, and you can buy these notes. And I recommend that you buy these notes because I make a lot of drawings and a lot of diagrams, and I would like for you guys to annotate using the lessons while I'm doing them. Okay, so you can buy these notes by contacting through the WhatsApp link provided below. And if you want to get private coaching for your specific needs, you can contact us right now using the WhatsApp link below. Okay. And have fun, you guys. So I'll see you in the next one.